First, see the cell cycle. Cell cycle, if you see, it consists of two phases. One is interphase, another is M phase. Interphase consists of three phases: G1 phase, S phase, and G2 phase. Now, G means gap. So this is first gap phase. First gap phase. So this is second gap phase. S means synthesis. So this is synthesis phase. If you check in the diagram, G1 phase is coming after M phase. So it is also known as post-mitotic gap phase. So G2 is known as pre-mitotic gap phase. Clear? Now, during interphase, we don't find any cell division. We are finding that only chromatin is there. Nucleus, you can study it. You can see the nucleus, you can see the chromatin, which can be stained with the basic dye, but you will not find anything else. But if you see, most metabolic active phase is actually interphase only. The interphase, though it is appearing to be resting, because cell division, when we study cell division, we should observe cell movement and cell division should occur, but we are not finding anything. So it appears to be resting phase, but it is most metabolic phase. And it is most metabolic phase. Here the cell is preparing itself for the cell division. Now let us assume that in the G1 phase, if suppose my chromosome is this diagram. First of all, you can't see the chromosome, but I'm just explaining, let us assume we are able to see the chromosome with one chromatid in the G1. Then what is happening in the S phase? In the S phase, see beta, analyze my diagram very carefully. They are busy in making the second chromatid. And to make the second chromatid, it means they should replicate the DNA or not. They should replicate the DNA. So see, I've drawn only one chromosome. Chromosome is one, but they are busy in replicating so that they can make their second chromatid. It means they can make the sister chromatid. So in G2 phase, you will find, actually if you will study, then you will find this chromosome. So by the S phase, the chromosome number is only one, but DNA content is double. Sister chromatid is made, and you know very well, but a sister chromatid will separate during anaphase. So can I say that S phase is the one in which DNA content is doubling as well as chromosome duplication is occurring? Can I say this or not? Very important line. S phase marks DNA replication and chromosome duplication. Why I'm using the word duplication? Because you have studied mitosis. It's not this, it is new topic for you. You know, okay, during metaphase, chromosome will come in the equatorial plate, and during anaphase, sister chromatid will separate and move toward the respective poles. And sister chromatid future, they are known as chromosomes, daughter chromosomes. Sister chromatid will behave like a chromosomes. So during S phase, we mark the DNA replication and chromosome duplication. Let me show you this line. Wait. First say. So see beta, during the division of the cell, DNA replication, cell growth will be there, cytoplasmic growth will be there. Okay. All this will be taking place in a highly coordinated manner to ensure correct division and formation of progeny cells. Now sequence will be there by which the cell duplicates its genome, means its genetic material, means its DNA, synthesis of other constituent of the cell, and eventually dividing it into two daughter cells. Then we say it is cell cycle. Cell growth means what? Cell growth, we are saying that cell growth. What does cell growth means? Cell growth means cytoplasmic growth. Cell growth means cytoplasmic growth. And it is a continuous process. DNA synthesis will occur only in the specific phase. During which phase it will occur? Only in the S phase. But cell growth is continuous. Again, it's a very important question. The replicated chromosome means duplicated chromosome are then distributed to daughter nuclei by the series of events, which will be under the M phase. And these events are also under genetic control. We'll be explaining you. Up till here, you are understanding it. Now see beta, see. First of all, see cell cycle. We are taking it in case of for human, it is dividing once in every 24 hours. In case of yeast, human is over. Okay, please learn this much. Then yeast. For yeast, cell cycle is 90 minutes. Clear? Now we studied, right now we studied the human cell will take how much time for the cell division? 24 hours. It is significant to note that in 24 hours duration of cell cycle of human cell division is only for an hour. 95% is taken by the interphase only. The interphase is taking more than 95% and only 1 hour is for your M phase. Clear? So we are saying the cell cycle is divided into how many phases? Cell cycle is divided into 2 phases, interphase and M phase. M phase means is the phase where nucleus will be divided, cytoplasm will be divided. Nuclear division we say karyokinesis, cytoplasm division we say cytokinesis. So M phase is a phase when the actual cell division occurs. The interphase is one which is present between the two M phase. M phase is one where the nuclear division will be occurring. You will say karyokinesis. And finally, the division of the cytoplasm, you will say cytokinesis. I told you when I was explaining on the board, the interphase is called as resting phase, but it is a time when the cell is preparing itself. So it's metabolically more active phase. Now, during G1 phase, the cell is metabolically active, continue grow, but it does not replicate its DNA. S phase, period during DNA synthesis or replication takes place. DNA will be doubled. Let us assume if suppose I am saying a cell is having 10 picogram DNA. After S phase, how much it is having? You will say 20 picogram. And if I am saying cell is having 10 chromosomes, after S phase, you will say 10 only. Is it clear? So basically, just 1 minute, beta, you can remember this way. Suppose, see, for learning this, best choice is this diagram only. Draw this chromosome. In the S phase, do this work, dot, dot, dot. It means it is not completed. And in the G2 phase, draw this. So how many chromosomes you are drawing? Chromosome is only one you are drawing every time. But how much DNA? Suppose here DNA is only one strand. Means it's a DNA one molecule. Let us assume one molecule of DNA. Means 2 strands are there. So here one is becoming into 2. And here finally 2. Clear, beta? That is why they are saying it. What is going on? In the replication, amount of DNA content is double. If the initial amount we say 2C, then it is increased to 4C. There is no increase in the chromosome number. If the cell is having 2N, even after S phase, the chromosome will be 2N only. I hope this much is clear. In animal cell, during the S phase, DNA replication begins in the nucleus and centriole will be duplicating in the cytoplasm. Why we are duplicating the centriole? So that they can make the spindle apparatus. And if they make spindle apparatus, it means we can separate the sister chromatid. Is it clear? It means spindle apparatus are also made from the microtubule. Means they are also made up of tubulin protein. During the G1 phase, protein are synthesized for mitosis while cell growth continues. Is it clear, beta? Now see here. Let us go to the last page, summary page, then I'll show you it. Cell cycle is divided into 2 phases called interphase, a period of preparation for cell division. Second is mitosis, means M phase. Generally, the student asks, M means meiosis also or what? Okay, it is meiosis. But our NCRT, when it is saying M phase, it is emphasizing for mitosis phase. So M phase is the phase where actual cell division is occurring. Is it clear? Interphase is further subdivided into G1, S and G2. Now G1 phase. G1 phase is the period when the cell grows and carries out normal metabolism. So cell grow means cytoplasm is increasing. I think so. You know, nucleus should be big in size, cytoplasm should be small. Nucleocytoplasmic ratio should be high. But now here ratio is getting disturbed. It means cell is preparing itself for cell division. Most of the organelle duplication also occur during this phase. Please learn this statement. Most of the organelle duplication occur during this phase only. Now come to the S phase. S phase marks the phase of DNA replication and chromosome duplication. I was trying to explain you this word only. Chromosome duplication, we say. Is it clear? It is nothing making of second chromatid, making of sister chromatid is going on, because that will only separate and behave like a daughter chromosome. Now G2 phase. G2 phase is the period of cytoplasmic growth. Though it is a continuous process, cell growth is a continuous process. Is it clear? I think so. This word is only very important, which I wanted to highlight to you. Ki chromosome duplication, when in the paper it is coming, the students are getting confused because they never think in this way. The sister chromatid which is going to separate, that is why they are calling it as chromosome duplication. Is it clear? Number is not increasing, number is same. DNA content is doubling, chromosome duplication is going on, and this work is going in the nucleus, and in the cytoplasm, what is going on, beta? Centriole duplication is going on. Centriole duplication is going on. So I think so interface from here, every point, each and every point for your PMT question is clear to you. Now let us start with the M phase. This much is clear, beta. You can note it down, it will be helpful. And whenever if you are confused, please draw the way I have drawn it, dot, dot, dot. You will not do any mistake also. Now see beta next. How the centriole duplication. Okay, before centriole, let us first start with the M phase. Achha, if suppose the cell is not ready to divide, then what will happen? Because many cells will go for differentiation. They have become specialized cell, liver cell, stomach cell, intestine cell, skin cell, they have become specialized. So what they will do, they will exit this cell cycle. They will exit the cell cycle and they will enter into G0 phase. See beta NCERT. Some cells in the adult animal do not appear to exhibit division. They are not dividing. Heart cells and many other cells divide only when occasionally it is required, just to replace the lost part. These cells do not divide further, they exit G1. It means after telophase, they will come cytokinesis. Cytokinesis, they will enter into G1, but from G1 to S, they will not move. They will exit G1. Again, PMT question. What they are giving it, after telophase, it is entering into G0. After G1, they enter into G0. Where they will enter? Now see here, they exit G1 and enter into inactive phase G0. Now see carefully, beta. Here in our NCERT, see here. From after G1, first of all, they'll come into G1, and from G1, then they will, if they don't want to cross, then they will exit G1 and they enter into G0 phase. Please learn it properly. It's very important, beta. Cell in this stage may remain metabolically active, but will not divide unless called on. So, is it clear? Now, in animal cell, who will do the mitosis? In animal cell, mitotic cell division is only seen in the diploid somatic cell. In the diploid somatic cell, only you will see the mitosis. But in the plant, you know very well, plants are having two types of body, haploid body and diploid body. So in case of the plants, mitosis is shown by both haploid and diploid body. In plants, again question, the plants can show mitosis division in both haploid and diploid cells. Please learn this. This is again very important. So in animals, it is shown only by the diploid somatic cell, and in plants, it can be shown only by the, it can be shown by both haploid and diploid cells. Now, exceptions are always there. If you know, in case of the animal honeybee, males are haploid and they develop parthenogenetically. So exception, if you see, exceptions are there where haploid cell divide by mitosis, example, male honeybee. Is it clear? So now we have to start with the M phase. First line, again question area, which is the most dramatic period of cell cycle? M-phase. Because here full dramatic changes will be occurring. Nucleus will disappear, nuclear membrane will disappear. Instead of nucleus, you will find chromosomes. And how the chromosomes are made? It means chromosomes are made by the process of condensation. So during prophase, entire prophase, condensation is going on. Chromatin is getting condensed itself to form chromosome. So prophase, which is the first stage of karyokinesis, it is marked by the initiation of the condensation of material. Chromosome material will become untangled during the process and they will form the condensation will be done to form the chromosomes. In the meantime, the centriole, which has already been duplicated, means pair of centriole, means centrosome, which has already duplicated during S phase in cytoplasm, will now start moving to the opposite side. Now see the centrosome, which has already undergone duplication during S phase, now begins to move opposite pole. Now see the diagram, beta. This is your cell. Okay, I'm focusing only on the centrosome right now. This is your pair of centriole. They have already duplicated. Around this is your pericentriolar satellite, also known as mesule. I'm using one this color, another this color, try to understand. In which the microtubules are inserted. Microtubules are inserted. Slowly, what will happen? They start moving to the opposite side. I'm not focusing right now on the chromosome. Try to focus only on the centrosome so that you can understand the concept. This has reached here, and see, this is a star-like. Assembly of the microtubule will be done. From here also star-like. Assembly of the microtubule will be done. Microtubule assembly is going on. And now chromosomes. Here if you see, this is chromosome shape is taking. Okay, nuclear membrane has disappeared. I'm taking a cell with the 2 chromosomes. So nuclear membrane was disappearing and shape is coming on. During this phase, the chromosome has become properly condensed. It is a late prophase. And this proteinaceous disc-like structure, but remember, kinetochore on which the spindle fiber will be inserted. So from this side, I'm using blue color. It is inserted. Okay, and from this side, this color, try to understand. Now, if you see only this diagram, beta, focus only this diagram. Focus this part and this part, only this part. Means focus this only, this and star. I think so, isn't it ester? And ester is with the centriole, with the astral rays. So this is pair of centriole. And this is astral rays. The centriole with the astral rays is known as ester. How many ester I've drawn? 2. I will say amphiester. And I think so ester is, centriole is seen only in the animal cell. So in animal cell, the spindle apparatus is amphiester. And ester is coming because of centriole, so I'll say centric spindle apparatus. And they are making spindle shape. They are taking the spindle shape. That is why we say spindle apparatus, because the shape is coming like a spindle. Now see the NCERT. Centrosome, which has already undergone duplication, begins to move towards the opposite pole. Each centrosome radiates out microtubule called ester, 2 ester together with the spindle fiber called is known as mitotic apparatus. Please learn it properly. Is it clear? So in the prophase, what is going on? Chromosome material condensation is going on, and we find how many chromatid? one chromosome is having two chromatid. Chromosome is one, per chromosome, two chromatid. Two chromatid is there. One centromere, two kinetochore. I think so this much for prophase is clear to you. By the end of the prophase, you will find no Golgi body because nuclear membrane is not there. It means no ER, no Golgi, no lysosome vacuole, nothing will be there. Everything will be disappeared. So this is the thing which we observed in the prophase chromosomes. Here one chromosome will have two chromatid, and these chromatid beta, it is known as sister chromatid. Okay, it is known as sister chromatid with one centromere, two kinetochore, and 4 telomeres. And in the kinetochore, your spindle fibers will be inserted. Spindle fibers will be inserted. Clear? This labeling is clear. Now let us see, beta. This is your cell. Nuclear membrane was disappearing and the chromosome was appearing this way. I'll take a cell with two chromosomes. Your centrioles have moved to the opposite pole. Centrioles have moved. Spindle fiber are inserted onto the kinetochore. Few are running from pole to pole. See this, they are running from pole to pole, and few are inserted onto the kinetochore, which is present on either side. Is it clear? This is the end of the prophase going on and beginning of the metaphase. So what will happen? Slowly, slowly, the chromosome will come to align themselves on the equatorial plate. Now properly alignment will be done. They are coming on the equatorial plate. This is metaphase. Metaphase is making equatorial plate, or you say metaphase plate. Now after metaphase, what will happen, beta? First of all, in metaphase, if you see, I think so maximum condensation of the chromosome is achieved. Maximum condensation of chromosome is achieved. So it is the best phase to study the chromosome morphology. If you want to study whether it is what is a shape, size, everything you can study best in the metaphase. Now this chromosome which is running from this is spindle fiber which is running from pole to kinetochore is known as chromosomal fiber. This is known as chromosomal fiber. And the one which is running from pole to pole, it is known as continuous fiber. So two types of spindle fibers are there. One is running from pole to the chromosome. One is running from pole to pole. Now during anaphase, what will happen? Centromere will split and sister chromatids separate. Centromere split and sister chromatids separate. Now question, who is moving towards the pole side? Is it centromere or the arms? So I think the diagram is telling you centromere is moving towards the pole side. Arms are trailing behind. Is it clear? Arms are trailing behind. And the shortening. Why the centromere split? Because of the shortening of the chromosomal fiber. Chromosomal fibers are going for shortening, which is making the centromere split, and the sister chromatids are separating. I think so you are getting the shape. If you see in the diagram right now, chromosomes are assuming V-shaped. Why? Because I have drawn metacentric chromosome. To study the shape of the chromosome, best phase is anaphase. Anaphase is the phase where you can study the shape of the chromosome. Structure, metaphase, shape, anaphase. If you remember in the chromosome cell chapter, I told you metacentric V-shape, submeta, L-shape, acro, J shape, telo, I shape. So all the shapes which we study is during anaphase. Structure we study during metaphase. Now finally what will happen? Chromosomes have reached to the poles. Okay, and the cytokinesis is also done. I started with cell with 2 chromosomes and I made 2 cells each with again 2 chromosomes only. So telophase and cytokinesis I've shown in the same diagram. And you know how the cytokinesis is achieved in the plant cells? In the plant cells, we have already studied it is achieved by which method, beta? Cell plate method, which is made in the center and it is moving towards from the center, moving towards the periphery. Means it will be this way. Make this your diagram, imagine this way to learn it. Made from the center, move this way. Showing centrifugal growth. Centrifugal growth. And in animal cell, how the cytokinesis is achieved? By furrow method. Means make the cell this way and make it move this way. It is showing centripetal growth. Draw this diagram, it will be helpful for you. In the plant cell, make from the center, move out. The move out means centrifugal. And in the animal cell, you have to move in the center. It means centripetal growth. Clear, beta? So this is your mitosis. Now see the NCERT. Complete disintegration of nuclear envelope marks the start of the second phase. Again, they ask you the question. Please remember this is statement. It is not the end of the prophase. It is the beginning of the second phase. Condensation of chromosome is completed and they can be observed under the microscope clearly. This is the stage at which morphology of chromosome is studied easily. This only I try to explain. If you want to study the structure of chromosome, how many centromere, how many kinetochore, how many telomere, how many chromatids, best phase to study this only. Metaphase chromosome is made up of 2 sister chromatids. That is why numerical values also I gave you, which are held together by a centromere. Small disc-like structure at the surface of centromere is known as kinetochore. Proteinaceous disc-like structure, and they are the site for the attachment of a spindle fiber. Metaphase is characterized by all the chromosomes. What is the characteristic of metaphase? Please focus. Metaphase is characterized by all the chromosomes coming to lie in the equator with 1 chromatid of each chromosome connected by its kinetochore to specific fiber from one pole, and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. Why I'm if you remember, I focus beta. How many kinetochore per chromosome? 2, because they are saying it once again. See beta, with 1 chromatid of each chromosome connected by its kinetochore to one pole, and sister chromatid by its kinetochore to the opposite pole. So this diagram, that is why we have to explain properly, because they can frame the question from here. So this is 1 kinetochore, second kinetochore, 2 sister chromatid, 2 kinetochore. It is attached to this pole and this is attached to this pole. And they are coming in the equator. We call it as metaphase plate. Spindle fiber attached to the kinetochore, they move to the equator of the spindle and get aligned along the metaphase plate. Is it clear? Now, if they ask you the question, beta, please remember, this is your early prophase in which you can't see properly sister chromatid. Please remember. In the late prophase, sister chromatid is visible, but still not properly clearly visible. This is when they give you this diagram, it means transition is going on to metaphase. They have to come in the middle. Till now, they have not come, but they have attached. See, they have attached. Okay, 2 kinetochore. Focus the diagram. You can check carefully. 2 kinetochore. They have attached to the kinetochore. So this is a transition when they will come back in the. Sorry, they will come exactly in the middle of the cell. Then we say this is metaphase. Clear, beta? Each and every diagram is important. And if you see here, this is your astral rays. These are astral rays and this is centriole. This is centriole, and around the centriole astral rays. That is why we say 2 ester are there. Now anaphase. During anaphase, each chromosome arranged at the metaphase plate split, and two daughter chromatid, now referred to as daughter chromosome. That is why in the S phase we say duplication of chromosome, because sister chromatid are now known as daughter chromosome of future daughter nuclei. They migrate to the opposite pole. Now, as each chromosome moves away, centromere remain directed towards the pole. Hence, at leading edge, centromere is leading and the arms are trailing behind. Arms are trailing behind. Thus, anaphase is characterized by splitting of centromere and sister chromatid separate. Now, in anaphase, if suppose they ask you question, I started a cell with 10 chromosomes. In anaphase, how many chromosomes are present? Your answer will be 20. Reason, now see, in metaphase, how many chromosome diagram is showing you? 1, 2, 3, 4 chromosomes. But see in the anaphase, how many? 1, 2, 3, 4, 5, 6, 7, 8. Total 8 chromosomes in anaphase. Because till now I have not started telophase. I have not started telophase, beta. Is it clear? In anaphase, actually, double. Double, I will say number will be double. Because now these sister chromatids are known as daughter chromosomes. Again, beta, it's a question. Please remember this way, they'll ask you. In telophase, what is happening? Chromosome reach to the poles, they decondense, lose their individuality. They are no longer seen and each set is now reached to the poles. The chromosome cluster at opposite poles, identity is lost, nuclear envelope reappear, nucleolus, Golgi and ER is made. Now, if suppose who is making the nuclear envelope? I told you the PMT question, rough ER will make nuclear envelope. Because nuclear envelope give rise to rough ER, so rough ER is only making the nuclear envelope. Up till here, clear, beta? Now, cytokinesis. Division of the nucleus. Mitosis, not only segregation of duplicated chromosome, you see duplicated chromosome here, into daughter nuclei means not only karyokinesis is done, but even the separation of cytoplasm means cytokinesis will be done. And how it is achieved in animal cell? In animal cell, it is achieved by the appearance of furrow, which gradually deepens and ultimately join in the center. So it means furrow start from the periphery and moves to the center. This is centripetal growth. In case of the plants, see the plant cell, because they are having the cell wall which is inextensible. Now it will not go for extension. Therefore, they go cytokinesis by different method. Which method? Wall formation start in the center. They start in the center by a remain of spindle fiber coming in the center. Now again question, I'm saying remain of spindle fiber come in the center. Vesicle of Golgi are strengthening it to form middle lamella. So beta, tell me one thing, cytokinesis in the plant cell is achieved by tubulin protein or not? Logically, can I say cytokinesis in plant cell is achieved by tubulin protein? Because remain of spindle fiber is what? Made up of microtubule. Microtubule is what? Tubulin protein. Can I say logically or not? And beta, in the animal cell, right now I was explaining you that cytokinesis means what in the animal cell? Furrow appear in the plasma membrane. And who is giving strength to the plasma membrane? Microfilament. So can I say cytokinesis in animal cell is with the help of microfilament, but cytokinesis in plant cell is with the help of microtubule. And you know, microfilament means actin protein. Microtubule means tubulin protein. Is it clear, beta? And grow outward. So in the plant cell, cell wall formation start in the center, grow outward to meet the lateral wall. Formation of new cell wall material begins with the formation of the precursor known as cell plate. And cell plate is the one which represents the middle lamella. At the time of cytokinesis, organelles like mitochondria and plastid get distributed between the two daughter cells. Sometimes what happened, karyokinesis is done, but cytokinesis is not done. So it leads to the multinucleate stage. Multinucleate stage in the fungus is known as cenocytic. I think so in the phycomycetes we studied. Cenocytic mycelium means multinucleate. Multinucleate condition in the plant, we say syncytium. For example, liquid endosperm in coconut. If multinucleate condition is seen in the plant, we say syncytium. Multinucleate condition is seen in the fungus, we say cenocytic. Multinucleate condition seen in the slime mold, it is known as plasmodium. So I think so mitosis concept is clear to everyone. Significance, it is equational division because equal number, usually restricted to diploid cell only, and especially in the animal cell, because in the plant cell, haploid cell also divide. However, in some lower plants and in some social insects, male honeybee, we have seen already, haploid also divide. It is very essential to understand this division. It is result in the production of diploid daughter cells with identical genetic complement. Growth of multicellular organisms because of mitosis, and cell growth result in disturbing. If you know, cell growth means cytoplasmic growth. Cytoplasmic growth means cytoplasm is becoming big in size, nucleus is not able to increase. So nucleus control is disturbing. That is why cell decided to divide, because generally the cell prefers to be small in size so that nucleocytoplasmic ratio should be very good. They want high nucleocytoplasmic ratio. And when cell growth occur, then what happened? The ratio is disturbed. Which ratio? Between nucleus and cytoplasm ratio is disturbed. So what they do? It has become essential to divide so that nucleocytoplasmic ratio come back. So healthy cell means normal cell. What they want nucleo to cytoplasmic ratio should be high. But if this ratio is disturbed, and this ratio will be disturbed, why? Because of the cell growth. Cell growth means cytoplasmic growth. We have already studied cytoplasmic growth. Then cell divide. Is it clear? Second important contribution of mitosis is cell repair. You know very well our epidermis, lining of gut, blood cells, all are constantly being replaced. If you know, meristematic tissues are also dividing by the mitotic division only. Apical meristem, lateral cambium, they are responsible for continuous growth of the plant throughout the body. So I think so up till here, beta, mitosis clear. Now let us see the summary part. See, the mitosis is divided into 4 stages: prophase, metaphase, anaphase, telophase. Chromosome condensation occur during prophase. In the same time, centriole move to the opposite pole. Nuclear envelope and nucleolus disappear. Spindle fibers start appearing. Metaphase means alignment of chromosome at the equatorial plate. In the front, it is given metaphase plate, but exactly it means in the equator means middle of the cell, so we say equatorial plate. During anaphase, centromere divides, chromatids start moving. Once the chromatid reach to the poles, the chromosome elongation start. Chromosomal, once the chromatid reach to the poles, the chromosome elongation starts. Please, again, they can frame the question, when the chromosome elongation start. In the telophase, nucleolus and nuclear membrane reappear, telophase. The chromosome shortening in the prophase, chromosome elongation in telophase. So I think so up till here your mitosis is clear with.